Hey there, welcome back to Principles of Ecology. This is another lecture. This week we're going to be going into species diversity, community composition, and ideas of succession. So I'm going to share my screen. All right. The two readings for today for sort of housekeeping are this nature, education, knowledge overview of succession and what that is, and then one chapter from the Gotelli book of Primer and Ecology, and it's chapter 9, which goes over species diversity and the estimation thereof. And so I start, I think, talking about ecological communities, what diversity is, and then at some point switch over and start talking about, and so like sort of halfway through, maybe a little bit more, switch over to talk about community composition and community assembly. We'll see how much, I think we might be able to get through all of this in one lecture and then just go over more fine scale things in class, but I'll play it by ear. I may only go over the first bits of this to leave some just for class. Okay, so I'm trying to sort of build on our previous knowledge and what we've accomplished so far. And so we've talked about species niche, which is the set of abiotic and the set abiotic. Some people think biotic. I maybe should delete that. It's the set of abiotic structuring axes along which a species can persist and not persist. Sort of defining that multidimensional space that is the n-dimensional hypervolume. And so a lot of this has largely considered individual species so far, right? So we learned like species population dynamics, and only recently have started with the Lotka-Volterra competition model, really discussing multi-species communities. And then there's only 2 species. And so now we're going to talk about an ecological community more generally. And so first we should start by defining what we mean by an ecological community. And here I defined it as the set of interacting species which utilize the same environment. Typically, the interaction of individuals of these species is assumed. If an interaction is not present, sometimes people would refer to it as an assemblage instead of a community. I think it's somewhat esoteric. You're somewhat splitting hairs at that point. Normally, community is defined at a single trophic level. And so what I mean by that, because we'll talk about sort of trophic stuff later, is that the species are interacting and are it's not comparing like plants to animals, for instance. And so like all of the individuals are like within a band of sort of taxa or groups of species that are likely to interact. However, some people are more liberal with the definition of community, basically using it to encompass an entire sort of food web, which I put in italics here, which includes the entire sort of chain from like a bear to squirrels to plants and everything. That is a little bit too loose of a definition in my mind. I like the idea of defining it based on species that are likely to interact in a given area. So yeah, I think I like our definition, or the definition that I give it up front the best. They are found in the same place and they're an interacting set of species. All right. Okay. So here what I'm trying to get into is the issues involved in sampling species and trying to understand who is in the community, right? So let's say you have some set of species I'll use colored dots here. I'm going to change the green just in case because of green-red color blindness. So hopefully I can make points that are colorblind friendly. And hopefully these are such. Is there green-red color blindness? I don't know. Hopefully you can, sorry, blue-red color blindness. Hopefully there's a different, you guys can see the difference between those. Yeah. If not, yeah, feel free to let me know. I can redo this with different shapes or something. I can figure out how to do that. Okay. Where were we? Okay. I think here I'm trying to like really loop in a bunch of other ideas that we've gone over. So in some community, there can be 4 species. So if this was a community, I'm going to draw the bounds of a community. There's 4 species in that community. Let's say we have another community that we're drawing arbitrary bounds on. And it only has these 2 species. We have 2 species community here, 4 species community here. That makes sense. According to some of the theory that we've gone over, if these species are competing for the same limiting resource, whether it be space or some food or light resource, they all of them but one will go extinct. That's not what we see in natural systems. Typically, instead we see some sort of form of dynamic coexistence. But how do we start to quantify what we mean by diversity? And so if we were in class right now, and I may do this still in class later, is how do we start to define what biodiversity or diversity means and the different sort of scales of it? And so here, this is sort of species diversity at the community level. But you can imagine each one of these is actually, if we consider them individuals, a community could look like that and have the same number of species in it, right? It's 2 species. And so this starts to get at questions of how we define diversity. In a very simple way, we can define diversity as alpha diversity. This is also called species richness. This defines diversity as the total number of species, of unique species in a community. So here, sorry, I'm just gonna copy the entire thing. Here, the alpha diversity would be 4. Here, the alpha diversity is 1. Here, the alpha diversity 2. There are 2 unique species. But there's also gamma diversity. Gamma diversity is the regional diversity. So let's say we have these smaller communities here, and let's say this is a geographic space that these exist in, but it's broad enough geographic space to where we can draw bounds around the communities. For instance, this could be viewed as ponds in a landscape where they're kind of connected, maybe through like big rain events, but not really connected. We can consider gamma diversity. So we know species richness within the community, alpha diversity of this community is 4. Gamma diversity is the diversity of species across the entire region. And so gamma diversity here is 4 for everything. So species alpha diversity 4, alpha diversity 1, alpha diversity 2, gamma diversity 4. And it's a regional species pool. All right. We may also want to think about diversity in terms of the differences in diversity between sites, right? You want a measure that compares 2 communities and says, how similar are these communities? How different are these communities? And for that, it's called beta diversity. And that is inherently pairwise. So I can highlight this community and say, what's the beta diversity of this community? That question doesn't make any sense. Beta diversity is only defined pairwise between two communities, sort of fundamentally. Okay. Now that we like sort of understand what these different forms of diversity are, we have alpha diversity, beta diversity, gamma diversity. We can start thinking about how we actually go about measuring them. And so instead of talking about the concept as if they're completely abstract, we're going to talk about the sort of fun nuances in the measuring, in the measurement of things like alpha diversity. And so we can imagine a situation. I give an example of like jelly beans in a bowl where each jelly bean is an individual. How we sample and how we monitor ecological communities is not by sort of complete censusing. So censusing would be measuring and counting all individuals in a given area and actually having sort of an absolute knowledge of what species there and how many are there. And that would allow direct estimation of alpha diversity, of species richness. But we don't do this. It's cost prohibitive, right? Like you can't count all the blades of grass and all the different species in like a tropical rainforest plot. It's just not possible. And so what often we do is take a sample and we assume that this sample is representative. And so in this community or this community here, I may just go that. And so I just highlighted the box. That's my sample. This is what I think is a representative of this community. However, if I measure the alpha diversity here, we have two unique species, so alpha diversity is 2. And if I call this a community, so I'm going to copy a gray box over there. And now this is my subsampled community. And I count the number of unique species, and I only have one unique species. I am underestimating species richness. All right. Okay. And so that's not good. I'm underestimating my species richness, right? So ideally, we want to develop a way that we can robustly sort of get at species richness, even from a sample. And one of the sort of most sort of obvious things that could influence our ability to successfully quantify alpha diversity would be the relative proportion of of individuals of different species. Here's what I mean by that. I'm just going to copy this entire thing and move it down here. Okay. So let's say I have these two communities. All right, and they're both going to contain the same two species. Oops. Oh. Okay. They're both going to contain the same number of individuals, right? But one contains only one blue individual or some small number of blue individuals, and the other contains a more even representation between the two species. It will inherently be easier to estimate species richness or alpha diversity for this community, right? Because if I take a random sample, I'm more likely to sample both blue and this tan green color. Whereas here, if I randomly sample, I'm way more likely to just get this dominant species. And what one sort of thing that we can use to sort of diagnose if our sample or what the sort of shape of the community is like are rank abundance plots, which I'm just going to make one. All right, so this would be what these look like is on the x-axis, or sorry, on the y-axis, we have the number of species that have a certain bin of abundance. And then on the y-axis, we have, there it goes. We have the relative rank, or it's actually the number of individuals. Sorry, I'm thinking of species abundance distributions, which I don't think we'll go over. Excuse me. Sorry about that. So I'm looking at my notes from last semester, which I got to draw on paper. Yeah, and we won't go over species abundance distributions in this class. And so essentially, rank meaning we just order the species. Actually, let's just delete that. We order the species. So this is species 1, and this is species 2. And we rank them by their abundance. And so it's highest abundance. So this one corresponds to this, and this one corresponds to this. And this should actually probably be like even lower, right? Because it's one individual. All right, and so if we see this, we can and extend it into like a multi-species community, so more than 2 framework, we can start to see that the peakiness and the subsequent drop-off, like the steepness of this drop-off, can tell us something about how even the community is. And so measures of evenness try to get at this where some species like this greenish, I don't know what color that is, are very, very common, while other species like this blue one in this community are very rare. And commonness and rarities, I mean, like that's basically just like community ecology in a nutshell. It's all commonness and rarity. You have a couple very common species, and then you have a lot of very rare species. Tends to be it. All right, so then we're going to copy that. Just for some completeness, we're now going to make the rank abundance plot of this. And here it would look like that. Oop. Looking like a much more even community. All right. And now we'll talk about the sort of approach to actually how we estimate species richness. This is like a sort of brief aside to talk about evenness. But there's 2 main ways to estimate species richness, and that's rarefaction and species richness estimators, which is, yeah, it's confusing because like the process of species richness estimation, how would you do it? Species richness estimators. But yeah, so we'll talk about rarefaction first. Rarefaction is the idea that you can subsample your data. So subsample meaning taking a smaller portion of the sample. So we have our true community and we repeatedly sample it. And so that's one sample, and that's another sample, and that's another sample. That's another sample. And essentially we do so in a way that statistically we can start getting at the true diversity, because we can build up a curve. And so the idea here, there's multiple ideas. Let me actually see how I go into it. Okay, I differentiate between individual-based and sample-based rarefaction. So what I just defined as where we subsample our data in groups is called sample-based rarefaction. So that involves subsampling the data, seeing sort of what the diversity is in that subsample, and then sort of using it to interpolate the sort of diversity given some level of sampling effort. So here's what, or I'll discuss why that is important in a bit. First, I'll distinguish between that and individual-based rarefaction. So that would be sample-based rarefaction. Individual-based rarefaction is when you have information on individual occurrences, which is typically how the data are structured, right? So so you'd have, instead of subsampling the data by group or by batch, the data exists just as individuals. And so I know that I sampled in my trap, I'm just trapping like small mammals. In trap A, there was this blue individual. In trap B, on the first day of sampling, there was this individual. On trap C, there was this individual. So I have, I can go over that in more detail, especially with more sort of clear and cogent examples in the lecture to really nail that home, because they sound similar, but they're sort of slightly different, ever so slightly. And rarefaction is largely for interpolation and largely for experimental design. Not entirely, but largely, I'd argue. That's one way to think about it, at least. And so what I mean is, it's like saying, given that we sampled this community really extensively, what is the amount of effort that we would need to sample X percentage of the species that we already know are there? That would be a good application of a rarefaction-based richness estimator. That being said, you can also use rarefaction to get at the expected number of species in a community when you only have a smaller amount of data, when you have an incomplete census, when you don't have a census, you have a sample. And here I try to distinguish between those sample-based and individual-based rarefaction. And so I say experimental design consists of sampling individuals. And I gave the example of small mammal trapping at a site. Individual-based rarefaction is appropriate for that. All right. Yeah, and here's the bit where I talk about a little bit of extrapolation. So basically, like, we can try to get at the mathematical relationship between the observed species richness and the number of individuals that I've sampled. Normally, it's used to aid in experimental design, like I said, to interpolate, so to look at, like, I sampled N individuals, but I really care about what species richness I could get at by only sampling N minus 10 individuals. This is a way to sort of gauge sampling effort required to estimate true diversity. But you could use it to extrapolate. So interpolate, meaning like fill in before, extrapolate, meaning if I sampled 1,000 individuals, or like two times my sample size N, what would I expect to see in terms of species richness? And there's a bunch of assumptions that go into this approach, the rarefaction approach. Yeah, it sort of assumes that the community is constant over time. If you're unwilling to say, like, I estimated species richness it assumes a constant community over time. It doesn't really bake in many dynamics. It doesn't really account for immigration or emigration of species, including transients. So species that couldn't persist there, but just show up in a sample, like you'll sometimes see. It assumes a well-mixed community. Yeah. So there's no like microhabitat variation, or you're sampling microhabitats in a very equitable way. I think that's what it would assume. And then also that individuals are sampled at random. And so that in the small mammal case, it sort of makes sense. But you could imagine a case like, what if trapping efficiency was really, really good for mice, but not good for shrews or voles? They just hated traps. They wouldn't go anywhere near them. You could be, you'd be disproportionately sampling mice at a higher rate than you'd be sampling voles. And that could influence subsequent rarefaction and species richness estimation. All right. Okay, and then I sort of plop this little equation in here, which I think is taken out of the Gotelli reading, maybe. It seems like it would be. But basically, it's a way to approximate the expected number of species, that alpha diversity that you see, given that you sampled M individuals, which is something as like a field ecologist they'd really want to know. They'd say, like, how many individuals do I need to sample to get an accurate estimate of what the overall diversity, alpha diversity of a community looks like. And so here it's the true diversity, which you have to know. I guess you're estimating it as the number of species that you observed, given a sample size larger than M. And then discounted from that is the proportion of species in the original sample and raised to the power in terms of the number of individuals in the sample. Yeah, we might play around with that in the lab. There's some of these things that are a little bit kind of fun to play around with and a little more intuitive once, if you can directly interact with them or sort of see how they work. And by lab, I meant class, like in lecture. Yeah, I sort of plopped it. It's kind of cool, but we don't really need to worry about this. I'll go really fast now. So normally rarefaction is used for, it's one site. Like you go to a site and you say, I want to know how many species I could estimate, or how many species are present given that I sampled this many individuals, in the case of individual-based rarefaction, not sample-based where we're taking a sample, a batch sample. What we also can do is use rarefaction to compare species richness between sites. And how you do that is if you have two communities that you believe are similar enough, you can subsample the more well-sampled site. So you have two communities that exist close in space. You can bootstrap or say like, these communities are representative, right? So this community is like relatively unsampled. This community is very well sampled. If you subsample this large, more well-sampled community, you can start to get at, and that's related to this idea right here, right? So like if I only can sample 100 individuals, which is, let's say that's the sampling that I've done here in this other site. So given that I've sampled 100 individuals and I think these sites are equal, randomly pull 100 individuals from this site and then compare it to the observed richness in this site, if that makes sense. So that's a way to try to compare species richness between two sites that are unequally sampled. And if that's confusing, I'll go into more detail in the lecture on Zoom later. Sorry if that was confusing, but I think you got the gist. All right, and so now we'll talk about methods for species richness estimation that are not rarefaction based, and that are typically used for extrapolation, and that tie a little bit more into this rank abundance distribution and the sort of the sampling process itself to an extent. And so here I introduce what's called the Chow Index. It is named after its creator, Ann Chow, who's a community ecologist, population ecologist. And so the idea of the metric is that it's it assumes that you have a sample, right? That you come in and you sampled some portion of the individuals in a community, and it uses information on the tail of this distribution. So if I were to, I guess those are species then. If I were to sample this community here, that has 4 species, but I didn't know it had 4 species because I never observed this species, this yellow species. And I observed this one only once, and I observed these 2 a bunch of times. That tail, those individuals or those species that I've only seen once or twice start to get at rarity in that community. And so by leveraging information on the number of species that are singletons, i.e. those that were only sampled once or are represented in the sample by a single individual, it's more clear, or doubletons, which there's 2 individuals given my sample, we can start to get at the estimated species richness in the area. So here, est is the estimated species richness. That is the Chow estimate of species richness. This is what you observe. So here that would be 2. Because in my example earlier, because I said I know these are there, I sampled 2. And this one was a singleton. And so F1, that's the number of singletons, and then divided by 2 times the number of doubletons, where F2 is the number of doubletons. So in my example, that would be 1 squared divided by 2 times 0, because there's actually no, so that's actually undefined. That's a bad example then. But yeah, sometimes in simple examples, they fall apart. But yeah, so F2 is doubletons, F1 is singletons. And it's a very simple relationship, but it's actually fairly informative and can be quite accurate despite its simplicity, which is odd. All right. These also bypass a lot of the same assumptions that, or a lot of the assumptions that Rarefaction made. But some of them remain true. Do you assume that individual? I think you still assume that individuals are sampled at random, but individuals can or species can differ in their detection probabilities, and the species richness estimator would still work. So maybe this one can be relaxed. You still do assume a well-mixed community. And yeah, you still assume the community is constant over time. That would actually be cool to sort of look at temporal changes in communities and estimate species richness and see how estimated species richness changes. I'm sure somebody's done that. But. All right, so now we're going to switch gears. So we talked about measuring alpha diversity. Now we're going to talk about measuring beta diversity. So beta diversity was defined as the dissimilarity between 2 communities in their structure. Okay, I'm just making sure that I have covered in my notes. I will go over rarefaction in more detail because I did so in the notes last year, and I think it was a sticking point for some students, and so we can go over that in more detail in lecture, in our personal lecture, interactive lecture, if you will. All right. So now let's talk about beta diversity. All right. For that, I'm gonna need one of these. And one of these. Okay. So here, now these circles, instead of representing species, I'll change their colors so that we don't think of them as species, maybe. Oh, that is a very bright green. Oh, we already had that green. That's still a species. Let's make it this orange color, if you can see that. And this one will be this. No, we already had a gray. Maybe a nice purple. All right. So these circles now don't represent species, they represent communities. And what we want to know, we want to compare these 2 communities and say how dissimilar or similar they are. This is sometimes also referred to as species turnover. Though species turnover makes more sense in a sort of temporal way. And we'll go over it. We'll maybe say it when we talk about succession, which I may shift to another lecture. No, I'll probably say it in the same breath because I think we're doing good. We're pushing along. Okay, so beta diversity, or one common measure of beta diversity, is called jaccard similarity. It's bounded between 0 and 1, where 1 means that the communities are identical, and 0 means the communities are completely dissimilar. Here's how it works. It basically borrows from set theory. Set theory, we don't even know that. Let's not get into that. It basically is the fraction of species that are shared between 2 communities. So here, if I draw a box around this entire thing, I can say these are all of the species, right? There's a shared component. There's this that are independent to this orange community. There's this set of species out here that are just only found in the purple community. And this overlap are the shared species. And so what Jaccard index is saying is that you take this shared portion in the center and you divide it by the total, the sum of A, B, and C. So A being only found in the orange community, B being the shared component, and C being those unique to species, the purple community. It's important that we don't double count species here, right? You understand what I'm saying? So we have, we count species that are unique to this community, the shared component plus the ones that are unique to purple, meaning the shared component is not like double counted. So it's basically the number of unique species across both communities. Okay. And then it's a shared portion as the numerator and then the total as the denominator. And so if you have two species that are identical, that would mean the intersecting portion is the same as the union there. So that's, this notation means intersection. This notation means union. And so that means that those communities are perfect, they're identical, and Jaccard index is 1. When you have two communities that are completely dissimilar, there's no overlap, there's no shared component, that means this intersection is 0 and this index is 0. Okay. And why we might want to measure things like community dissimilarity or similarity is because there's a lot of cool relationships that can sort of emerge, right? So we did not only just like looking at succession, because that's basically calculating beta diversity, but also things like the distance decay relationship, which is a very classic sort of attempts to understand how communities change as a function of space. The idea is that more geographically close communities should be more similar in their composition. And so let's try, no, I'm going to leave that up there. I'm just going to copy it. Let's draw this out or sketch this out. All right, and so this is a plot, and the x-axis is going to be the geographic distance between sites. And the y-axis is going to be similarity, as we defined it using the Jaccard index, which means 1, communities are identical, 0, communities are entirely different. What we expect here is a relationship that's going to look like that. I want to make it thicker. I know I can. There we go. Dial back a bit. That's prettier. All right. And then what this means, and so if you're, that's where each point here is a pair of communities. So every point is, or every community is actually represented a bunch of times in this plot. So you have a set of community, a set of n communities. Every pairwise combination of those n communities is going to be represented in this plot. If that doesn't make sense, we can go over that further in lecture. But this is what distance decay relationship suggests. And can I go over it? Yeah, I talked about the reasoning a little bit in here. And so communities that are closer together should be more similar because they share species, because when we think about species niches and an spatially autocorrelated environmental landscape, nearby areas are more climatically similar, such they should have more similar communities. And that's one of the potential reasons for the geographic distance decay relationship. Dispersal limitations could dispersal limitation could also play a role, but I think the main sort of cause of this is having a spatially autocorrelated environmental landscape, in my mind. But feel free to disagree. We can talk about it. And the last little bit of this lecture before, maybe I think I will actually stop and do this as 2 different lectures just to give myself a break. And last little bit of this one is thinking about how alpha, beta, and gamma, I guess not really gamma, diversity have changed over time. And so there's a paper from 2014 in Science, I link here, basically show that there's little evidence that we're losing alpha diversity, which is really heartening. It's like, oh, nice, it's fantastic. So species richness as measured across different communities didn't really change that much over time. However, there was something else that was going on here, and there was an increase in the similarity of communities. And so this beta diversity, basically what is happening is communities were becoming more similar to one another, which means that we were losing species, we were losing rare species. But species richness was offset by the addition of widespread or invasive species. And so what we didn't see when looking at alpha diversity made more sense when we considered beta diversity. And so it's a sort of reminder that these different components of diversity sort of may require you to, or the researcher, to consider them jointly or together. Okay, and with that, I'm going to take a short break, and then I'll come back and we can talk about some community composition and assembly stuff. Should be fun times. So yeah, take care.